Welcome to AS London Wellness Academy. This video unit is looking at the structure of cells. Now, cells is the structure and functional unit of all living organisms. Some organisms, such as bacteria, are unicellular because it consists of one single cell. Other organisms, such as humans, are multicellular as they have many different cells. Each one of these individual cells taking nutrients to convert these into energy. They carry out specialised functions and reproduce if necessary. Even more amazing is that each cell stores its own set of instructions for carrying out each one of these individual activities. Now, a cell itself is the smallest unit that can live independently, and they come in all different shapes and sizes. A group of cells form tissues, and studying the structure and form of cells and tissues is known as histology. We're going to start off by looking at the anatomy of the cell. We're going to look at the nucleus. Now, we're going to start with this because it actually plays a central role in the cell. Information is stored in the nucleus and distributed to guide the life processes of the cell. There are two types of structures found in the nucleus, and these are your chromosomes and your nucleoli. Chromosomes can be seen clearly during cell division, and they contain your genes. Now, your genes are the basic unit of heredity which are passed from their parents to the children, and the genes actually guide the activities of each individual cell. Now, cells are a living structure, and they move around and have the ability to change. So your nucleus contains your nucleoli, your chromatin, and your nucleoplasm. Now, your chromatin is actually loosely coiled strands of DNA. And just before cell division, the chromatin become coiled even tighter to form your chromosomes. Moving on to looking at the second part of the cell anatomy, we're looking at your cell membrane. They surround and separate the cell from its environment. The cell membrane allows certain materials to pass through or enter and leave the cell. Your cytoplasm is a semi-fluid found inside the cell but outside the nucleus. We're going to now move on to the mitochondria, and this is the powerhouse of a cell. The mitochondria provide the energy where it's needed for carrying out the cellular functions. Your endoplasmic reticulum is a network of membranes, cavities and canals. It helps in the transfer of materials from one part of the cell to the other, and it actually transports proteins throughout the cell. Your ribosomes are protein factories in the cells, and they're composed mainly of your nucleic acid, and this can make proteins according to the instructions provided by the individual genes. Your centrioles, these help in the process of cell division. They are paired rod-like organelles. And then you have your lysosomes. These are membrane-bound spheres which contain your enzymes that can digest your intracellular structures or bacteria. So we're going to be moving on to have a look more into the detail of meiosis and mitosis. So we're going to start off by your cell multiplication. Now, individual cells have fairly specific lifespans. Some types of cells have longer lifespans than others. During the process of growth and repair, new cells are being formed. The usual process of cell multiplication is called mitosis. There are two important factors that you will need to consider. From one cell, we get two new cells. The genes of the new cells are identical to the genes of the original cells. We have got different phases. We have the interphase, so your cells may appear inactive during this stage, but they are quite the opposite. This is the longest period of the complete cell cycle, during which DNA replicates the central divide and proteins are actively produced. For a complete description of the events during interphase. Your prophase, during this first mitotic stage, the nucleolus fades and the chromatin, so your replicated DNA and associated proteins, condenses into your chromosomes. Now, each replicated chromosome comprises of two chromatids, both with the same genetic information. Your microtubules of the cytoskeleton, responsible for cell shape, mobility and attachment to other cells during the interphase, disable, and the building blocks of these microtubules are used to grow the mitotic spindle from the region of the centrosomes. Then we have your prometaphase. In this stage, the nuclear envelope breaks down, so there is no longer a recognizable nucleus. Some mitotic spindle fibers elongate from the centrosomes and attach to your kinetochores. These are your protein bundles at the centromere region of the chromosomes where your sister chromatids are joined. Other spindle fibers elongate, but instead of attaching to chromosomes, they overlap each other at the center of the cell. We have our metaphase then, which is where the tension is applied to the spindle fibers and they align with the chromosomes in one plane at the center of the cell. Your anaphase, so your spindle fibres shorten, your kinetochores separate, and the chromatids, which are your daughter chromosomes, are pulled apart and begin moving to the cell poles. Your telophase is when the daughter chromosomes arrive at the poles and the spindle fibres have then pulled them apart and they disappear. We're going to look at your meiosis next then. So a human body cell contains 23 pairs of chromosomes. The gametes or sperm or eggs contain half the number of chromosomes, which is why meiosis is sometimes called your reduction diversion. Before meiosis begins, the chromosomes are copied exactly. The DNA of each chromosome is replicated to form 2 chromatids. They then arrange themselves into pairs. This both codes for the same characteristics and prepare for cell division. At this point, maternal and paternal chromatids can exchange bits of DNA to recombine the genetic material and increase the potential for variation. Moving on to the second part then, is when the homologous pairs of chromosomes then separate and move to the poles of the parent nucleus. For each of the 23 pairs, there is 50-50 chance as to which pole the paternal or maternal pair of chromosomes go. With over 8 million possibilities, there are many opportunities for variation. The nucleus now divides to form 2 daughter nuclei, each with a mixture of paternal and maternal chromosomes, but with half of the full complement of genetic material. So there's no pairs in this at all now. This is called your meiosis 1. Finally, the 2 daughter nuclei themselves divide to form gametes. The second division, your meiosis 2, works like mitosis. The chromosomes, really pair of chromatids, split apart to form the genetic material of the 4 new cells, and their end result is 4 sex cells, each with a complete but single set of 23 chromosomes. On fertilization, the nuclei of the sperm and the eggs join to form a new nucleus called the zygote. Now this contains 23 pairs of chromosomes, 23 single chromosomes from the sperm and 23 single chromosomes from the egg. Now it's important that you don't get the two confused. So mitosis purpose is to make daughter cells identical to the parent cells. Meiosis is to produce sex cells. Mitosis takes place in all cells apart from the gametes, and meiosis is in the reproductive organs. Your mitosis produces 2 daughter cells, and your meiosis produces 4 gametes. Mitosis, you have the same number of chromosomes as a parent cell, and meiosis, you have half as many as in a parent cell.